All right, so I'm in charge of number 1 and number 2, but of course we'll start with 1, obviously. All right, so with number 1, the question is, this concerns a human disease called Angelman syndrome, which comes out to be with the recessive A, and then a non-diseased person has a dominant A. So they give us the cross between a male as a heterozygous and then a heterozygous female. But they also tell us that they tell us that the paternal allele is silenced because of the imprinting that's occurred. So when we come to our Punnett score to try and figure this out, I put it in 2 different colors just because it's easier to visualize a bit. Our male up here is going to be silenced. So these are in red. And then our females, since it's not silenced, is going to be expressed in the phenotype. So when we come over here, this is what our ratio comes out to be. And because of the silence, sorry, I'll set my laptop down. I'll be going back and forth, so bear with me. Because these are silenced, we can basically just cross them out and look at the female alleles in order to determine the phenotypes. So we've got 2 dominant, which means we've got 50% are non-disease and 50% are Angelman disease. It's pretty much the same rules apply for B, and except this time it comes out to being, here, I'll cross these out just so you can see it. The females are all recessive, so therefore it's all 100% Angelman syndrome. B. And then, all right, C is a bit more complicated, C and D, because we're talking about the bar bodies and how many are found in each cell. So in C, we have the triple X syndrome, meaning that there's 3 X chromosomes. And we know that there's XX on each one, but in the problem, it says that 2 of them have been deleted. So if we kind of take this visual, cross those out, that leaves us with 1 exit left. And since we need 1 exit to be activated, we won't have any bar bodies, so it'll come out. Where'd my markers go?
Sorry, man.
You're good. I'm going back and forth. So it'll come out to be 0 bar bodies, because we need this one to be activated still. For D, the situation is with the T6 and our X-cyst. Like the coatings that go around the chromosomes. Except our T6 is not working, but our X-cyst is. So our X-cyst is the coating that targets the chromosomes for inactivation, and the T6 comes over and removes one of them so that one chromosome can remain active. But because it's not working, this is going to inactivate both chromosomes, and therefore we will have 2 bar bodies. Voila. All right. So that's it for number 1. Number 2, since it's mostly just vocab, we'll just kind of give our answers. So for A, we have the SRY gene is an example of holoandric gene. Then B, SMIC2 gene is an example of a gene found on both the X and Y chromosomes. C, for C, we actually have to do the Punnett square because it gives us cross, and we have to figure out the phenotypes. So kind of like we did with number 1, A and B, with there being silenced alleles. This time it's the maternal allele is silenced, so that is in red, male is in blue. And this is what the Punnett square comes out to be. And because the female is silenced, we won't be looking at these red ones, just at the blue ones, the male's alleles, in order to get our phenotypes, which comes out to be 50% dwarf and 50% normal, or 1-to-1 phenotype ratio. For D, we get, in the sex influence pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other. I can never say this right. So genomic imprinting is an epigenetic process that occurs during gamete formation while dosage compensation is an epigenetic process that occurs during embryogenesis. F, so it's talking about the double strand of RNA molecules and what two molecules make up form the two halves. You have T6 and your exist that form the double-stranded RNA. And then G, when the reciprocal crosses give identical results, the alleles that control the trait in question are located on the Y-linked or the Y chromosome. When reciprocal crosses give different results, the alleles that control the trait in question are located on the X chromosome or the X-linked gene. And that's it for number 2.
3 says a woman's father has OTD deficiency, an X-linked recessive disorder producing mental deterioration if not properly treated. The one's mother is homozygous and is unaffected. So the first question A is asking us for the genotypes of the woman's father. So we know that she has, that her father has OTD. So we first off need to find her genotype. So we know that she is not affected, and that her father is. So because it's X-linked, he's going to give her a recessive gene. So I put that as a lowercase t. And then the mother, because she's dominant, recessive, and she's recessive. She's homozygous and is unaffected. We know that, well, it's just going to be the opposite. So I put those as 2 capital T's, and so she's going to get 1X from the mother, 1X from the father. So her genotype is X capital T, X lowercase t. So the father's is X lowercase t, Y, and then the mother's is X capital T, X capital T. So dominant, homozygous dominant on that one. For B, it says, if the woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? So for this one, we've already figured out her genotype, which is over here. A man that doesn't have it, because it's X-linked, we know that he's not going to be like the father, so it's not going to be a lowercase t, it's going to be a capital T. So X capital T, Y, so obviously T, super simple. For the son that would have it, would have OTD, is this one. So it would be a 25% chance that they would have a son with OTD. C says identify the genotype of a male with whom the woman could produce a daughter with OTD. So with this, we also know that it's 25% chance as well. So we just took her again, with the father would have to be affected in order for it to come through. Because if he wasn't affected, then it would be the dominant would take over and the daughters would be carriers instead. So with the father being affected by OTD, there would also be a 25% chance. Through there. So you have your dominant and recessive. Dominant, recessive, these would be the boys though, and these are the girls. And then D asked it says, consider the cross described in part C of this question. What are the expected genotypes and phenotypes of the offspring? So again, it's the same. So these ones, so the first one, they're going to be a carrier, but are not going to be affected. Carrier, unaffected, copper. See, yep. For the son is going to be just normal. For the girl, that's going to be, again, the one that is a carrier and actually has OTD. And then the next son is also going to have OTD. So that's number 3.
Okay, so I was responsible for number 4. That talks about the presence of a beard in some species of goat is determined by an autosomal gene that is dominant in males and recessive in females. So for a male, we've got big B is the dominant, little b is the recessive, females, little b is dominant, the big B is the recessive. So those will be our use. So for A, it asks, suppose that a cross produces a 3-beard, 1 no-beard phenotype ratio among male offspring. What are the genotype and phenotypes of the parents? So in order to get 3-to-1 with the males, we have to have dominance in 3 of the 4. So in order to get that, we had to use heterozygous both for male and female to get the one bearded male. Wait. Sorry, three bearded males, 1. I'm writing stuff all over the place here. Sorry. All right. 3 bearded males, 1 non-bearded male. For B, it says suppose that a cross produces a 1 beard, 3 no beard phenotype ratio among our female offspring. What are the genotypes and phenotypes of the parent? Oh my gosh, I was right. Hold on. My apologies, everyone. I'm correcting myself as I go. So, we have a 1 bearded male, a 1 non-bearded female for our parents. So, let's go back to B. So, for the parents of that one, again, we're going to be heterozygous. Reason being is that will provide 1 bearded female and then the 3 non-bearded females. And that gives us our female will be non-bearded heterozygous, the male will be bearded heterozygous also. For C, it says suppose a cross between a male with a beard and a female without a beard produces a 1 beard to 1 no beard phenotype ratio among female offspring. What are the genotypes of the parents? So in order for us to get a bearded female in some way, shape, or form, we have to have we have to have, in a 1-to-1 ratio, we have to have the mother, the female, as homozygous recessive for her technically, and then a heterozygous male, because this gives us a bearded female, and then a non-bearded female, So ratio is technically 2-to-2, is down to a 1-to-1 ratio. For D, it talks about suppose a male and a female, both beardless, produce a 1 beard, 1 no beard ratio among male offspring. In order to get that, we had to have a female with a heterozygous geno and a male with a homozygous recessive geno, because it creates, again, 2 beards, 2 no beards, down to a 1-to-1 ratio for the males. That's it.
Teaching number 5. So number 5, A and B are about cats. It says in cats, orange coat color is determined by the little b, and black coat color is determined by the big B. So the heterozygous condition produces a phenotype known as the tortoiseshell, and that these genes are X-linked. Suppose you cross an orange male cat, which we have right here, with a tortoiseshell female cat, which we have right here. What are the genotypes and phenotypes of the progeny? So right here in our Punnett square, we have the genotypes. We have X big B, X little b, and we have X little b, X little b, X big B, Y, X little b, Y. So the phenotypes of these, we're going to get 1 tortoiseshell female, we're going to get 1 orange female, 1 black male, and 1 orange male. Now part B reads, male tortoiseshell cats are very rare. What is the genotype of a male tortoiseshell cat? Well, the issue with males is we only have 1 X chromosome, and it makes it really difficult to express 2 different genes when they're X-linked. So what we end up with is a cat with something known as Klinefelter syndrome. So our genotype is going to be X big B, X little b, Y. Now what Klinefelter syndrome is, it's when there's a mistake causing meiosis during telophase 2, and the homologous chromosomes actually end up not splitting in one of the eggs, and both those X's will meet with the sperm, and you get your XXY genotype.
There. For C on number 5, it says, in the fruit fly Drosophila, a mutation produces the scalloped phenotype, irregular margins on the wings. Assume that the scalloped phenotype is the result of an X-linked recessive mutation. The genotypes and phenotypes the progeny produce with reciprocal crosses are done. So what we have is we have X capital T for normal and X lowercase t for scalloped. And I accidentally did C over here, so we have X capital T with the male. This is how you do reciprocal crosses. So you're going to have 2 normal, and the males are going to be the ones that are scalloped, whereas the females are actually going to be just carriers of that. And then it says for D, this question concerns the scalloped phenotype described in part C. Assume that the scalloped phenotype is the result of an autosomal recessive mutation. Indicate the genotypes and phenotypes the progeny produce when reciprocal crosses are done. So without the X chromosomes, because autosomal means that it's not sex-linked, so it's actually just going to be capital T and T, and then lowercase t. So with reciprocal cross, we're still going to have these on there. So again, you're going to have 2 that are carriers, and then 2 that are carriers, and then 2 that have scallops.